All right, so I think before I get started, so let me first thank the organizers, I think, for putting together a fantastic workshop. In particular, Chetan and Shantanu for the opportunity to participate. It would be remiss of me if I did not thank the student organizers who have done a fantastic job in terms of all the operational and the logistical support they have provided, without which I don't think this workshop would be the same. So thanks so much. So over the last 3-4 days, we heard a lot about vision. There was a lot of monkey business. So I think it's time to look at something new, different, right? So I think I'm going to focus on the sense of smell, olfaction. So we use very simple models. We use grasshoppers and fruit flies to examine how information is processed in the simple circuits, what are the biological design and computing principles of an olfactory system. And the hope is that if you understand the biology well enough, we could make an electronic nose, a non-invasive chemical sensing tool. The idea has been proposed for 30 or 40 years, but nothing works in realistic environments. So the idea is to see how exactly biology deals with challenges that actually you would have to solve in order to create a practicable device. So a majority of things that we do in my lab are centered along these 4 questions. So we are interested in how a very small insect brain performs pattern recognition. And there are multiple components to this here. So the first thing is how external cues, information about the external cues, are transduced and represented in the insect brain. So once this information is internalized, what are the neural representation associated with those external stimulus? And then how does the neural activity drive different kinds of behavior? And then finally, there is the engineering component. If we really do think we understand the biology well enough, can we create devices that match their capabilities? So a lot of things that we do, everything that we do fits within this particular umbrella. And this is a quick outline of today's talk. So I'm going to, again, I assume that you guys did not have a background in insect olfaction, so I'll give you a quick intro on insect olfaction. So I'm going to show you, a couple of days back, Professor Arun showed you how to actually perturb the visual stimulus in the spatial domain by perturbing CAPTCHAs. I'm going to do the same in terms of perturbing the olfactory responses, but using the temporal dimension. And then I want to see how robust odor recognition is achieved in a very simple model organism. How are these neural signals then mapped onto behavior? And then the work that I'm presenting has already been published, so if you want more details and methods, and even the data I think I've shared with some of the work groups. So you are welcome to try, and actually I have an explanation of what is happening. If you have a better explanation, go for it. All right? So before I talk, I just want to give you an intuition. Okay? A very simple example that we all understand, and then I'm going to tell you exactly the same thing in neural terms. Right? So let's start with a very simple problem of recognizing an object. In this particular case, the problem is to recognize a chair. There are so many different approaches. I'm going to propose one simple approach. Okay? The simple approach is as follows. We are looking for salient features. Okay? There are 4 legs, there is a seat, there is handrest, there is back support. If these 4 features are present, then that particular object that we are seeing in an image, we call it a chair. Okay? It's a very simple rule, correct? This works perfectly fine for this instantiation of a chair. But if you look at different instantiation of the object called chairs, not all of these features are present in all of these different objects. Some chairs don't have a back support, they don't have a hand support, some don't have 4 legs. With respect to all these objects, we all agree belong to this particular category called chair. So one simple way to actually achieve robustness and recognize all these objects as chair is to lower our constraint that all the 4 features have to be present. If we say any 2 out of those 4 features are present, then as you can clearly see, all of these objects satisfy that particular constraint. Would you agree with that? Does it make sense? So this kind of a concept in machine learning is called as a M of an N concept, meaning that any M out of the total N features uniquely identifies a specific class. We cannot guarantee which of those features are more important or more consistent, but as long as some combination of those features are present, that object is going to be there. Okay? So another way to think about this is this logical operation, disjunction of conjunction, or OR of ANDs. What do I mean by that? Feature 1 and feature 2. or feature 2 and feature 3, or feature 3 and feature 4. Any of those combinations is going to be sufficient to allow us to recognize that particular image that we are seeing as a chair. So far good? Everybody on board? Okay. So if you're, yeah?
There was one feature that was common in everybody.
Absolutely, yeah. So there are some, again, it's just a schematic example. Of course, there are going to be some features that are more robust. So without a seat, maybe there is no chair. So maybe there are some features that are more important, yeah? But in general, but to actually have the robustness, we cannot be too restrictive and too fixed on how we decode. We need to be more flexible. That's basically the message. All right. So in the next 40 minutes or so, I'm going to try to convince you that the way I just presented the object recognition is exactly how we can decode information about odorants from the insect brain. All right? I'm going to show you that only a flexible subset of neurons respond to a specific stimulus. We cannot guarantee any particular neuron is going to respond to an odorant all the time. Therefore, we need to be able to use flexible subsets of them to decode information. And finally, this operation, as I already told you, is a very simple logical operation, a disjunction of conjunctions. Okay? All right. So some background in insect olfaction. So this is the model organism that I work with, grasshopper or a locust. So you can see a front-on picture here. The two antennae, that is basically their chemical sensing organ. So in humans, you have your nose. At the roof of your nose, you have olfactory epithelium. You have millions of neurons there that actually transduce chemical information into an electrical signal. In the insects, it is the two antennae where there are olfactory sensory neurons. Each of these antennae has about 50,000 olfactory receptor neurons. So if you think about making a chip with 50,000 sensors, it's still, again, a pretty humongous, daunting task, right? And there are, in this particular organism, there are about 150 different types of sensors. So the set of proteins that they express, which they use for transducing the chemical information, that determines what type of sensor it is. And there are 150 types of them, right? There are several copies of the same type of sensors as well. And that is distributed along the length of the antennae. If you remove the small region, the exoskeleton or the cuticle between the two antennae, we can expose the olfactory circuits that are present that process the information coming in from the antennae. Okay? So this is a picture. This is a more schematized version of that entire olfactory circuit. The stem-like projections that you're seeing at the bottom right, that is supposed to schematize the insect antennae. Each color dot here represents one olfactory receptor neuron or one sensor. The color of the dot represents the type of the sensor that comes from the proteins they express. Okay? So there are multiple colors and multiple copies of the same color as well. The other thing that you can see here is that the output wires, their axons, are amazingly very well sorted such that all the dots of the same color, all the olfactory receptor neuron that expresses the same set of proteins, they send their output wires to the same region, to the same part in the next center, which are spherical structures of neuropil called glomeruli. Okay? You can think of them as olfactory channels. In the locust, there are about 800 of them, right? And this particular circuit is called the antennal lobe, which receives information from the senses directly. And here there are two different types of neurons. These are cholinergic or excitatory projection neurons. These are GABAergic or inhibitory local neurons. These neurons fire action potentials. These neurons do not fire action potentials. They are more analog, if you wish. Yeah? So the interaction between these two neural populations transforms the sensory input that is coming in from the antennae into patterns of spiking activity that is patterned over space and time before they are sent to higher centers such as mushroom body or lateral horn. Okay? So it's a very simple schematic. 3 hops, you're already there in the memory and the learning center, which is also true for humans too. Your nose, your olfactory bulb, olfactory cortex, 3 gems, you're already there. So the shortest path to your brain is through your nose. Yeah? So far good? Yeah?
So in the 150 protein types you have,
yeah,
do you consider these like redundant or kind of orthogonal, or we shouldn't think of it like that?
So it's not a lock and key mechanism. So there is some overlap between what each sensor type picks up. That's a combinatorial code, combinatorial input that comes from the antennae to the following circuits. Any other questions? Yeah?
So where are the cell bodies? The way you've drawn, it seems that they're somewhere else.
Yeah, welcome to the invertebrate world. Yeah, so the cell bodies are always away from the place where the action happens. These are unimodal neurons. They sit in the cluster separately. They send their processes. The processes branches, and one side goes to a dendrite, The other one goes to becomes an axon. So only the projection neurons have the axons. Local neurons do not. So in fact, there are 800. So these are 800 neurons on the left side and the right side of the insect antennae, on the insect antennal lobe. If you remove these neurons, the locust is as good as not smelling anything, anosmic, right? So this is all the information that that particular organism has about any volatile cues that are present in its environment.
But is it in the antennal lobe?
It is in the antennal lobe, but sitting on the periphery, on the edge, separately, yeah.
What activates these projection neurons or local neurons?
So the sensory input that is coming in from the olfactory receptor neurons, these are the sensors. They convert the chemical information into electrical signal, which are then sent here, which are then picked up by these two neural populations. Does that make sense? They are directly driven by the sensory input.
It looks pretty interesting from this architecture point of view.
Yeah.
You have a large number of inputs are coming in, going to the lower dimension, then from lower dimension to the higher dimension, like encoding architecture.
Yeah, so I think, yeah, it is funneling in and funneling out of information, as you really said. It is also a shallow network. Vision, we talk about deep neural networks. Olfaction is a shallow neural network. So I think it's different ways. The one other thing is different about olfaction versus vision, for example, it is a lot more higher dimensional. So there are any combinations of chemical features you put in it becomes a new odor, right? That's what the perfume industry is basing their life on. Yeah? Any other questions? Yeah?
In the mushroom body, do you think there are in the mushroom body, do you think there are like lateral connections or compartmentalized form of learning?
So I'm not going to talk anything about the mushroom body, but again, mushroom bodies, there are connections from giant GABAergic neurons, and there is also some interaction that comes from neuromodulators and things of that nature. So, but I'm not going to get into that aspect at all. I'm going to limit myself to only this particular neural population, projection neurons. Yeah? Any other questions? Okay, all right. So a lot is known about how odors are processed and how these different neurons respond to odorants. Okay, so I'm going to focus on this particular circuit called the antennal lobe, and this predominantly I'm going to focus only on this particular type of neurons, the projection neurons. So I don't know how well you can see this, but again, this is a picture of the antennal lobe under a microscope, under a compound microscope. You can roughly see some rough outlines of the cell bodies or spherical structures. Those are actually the soma or the cell body of projection neurons. If you look, if you squint your eye a little bit, you can also see this pipette that is coming in from the bottom left. So those are the patch clamp electrodes, which we go in, we apply a little bit of suction, suck the cell into that particular pipette, rupture the membrane, listen to what is going on inside the cell. Okay, that's basically intracellular recording. And if you do such a recording, what I'm showing you here are intracellular recording traces showing membrane potential fluctuation as a function of time. So we are measuring the voltage difference between inside of the cell and outside of the cell. And there are 7 different projection neurons from the same locus brain. X-axis, time. The scale bar tells you what the unit is, 50 millivolts, right? So this dotted line is when an odor puff was presented to the insect antennae, right? So what you're seeing here is before any odor comes in, these neurons are spontaneously have some activity. As soon as the odor comes in, you can see that some neurons becomes extremely strongly activated. Others are hyperpolarized, meaning they are shut down. And there's some combination of those, right? And after the odor pulse is terminated, the response does not stop there. You actually still have activity continuing, right? So some neurons get released from this inhibition. These are the on neurons, these are the off neurons over time. So in vision, everything is in space. In olfaction, everything is in time, all right? So you have on and off channels, on being when the neuron is active and off being active after the stimulus has been removed. So far good? Yeah?
So these are all getting the same channel fibers? Like they're coming from the same type of like the pink colored axons?
So they are getting input from different sets of olfactory sensory neurons. Yeah, they're not driven by the same input, yes. But some combination of those inputs. Okay? So feel free to stop me, all right? So I think, again, I know you guys are not used to looking data in this way. Yeah?
If you want to stop you,
yeah.
are there oscillations in this?
Oh, yeah, I'll show you that. Of course, there are oscillations. Very weakly gamma, if you want to say. No meditation, though.
As I said, like, the olfactory coding is all in time, but like as different projection neurons are active for different odor.
Yeah.
So isn't that in space too? Like,
so it's both space and time. Yeah. What I'm saying is that whatever perturbations or whatever way you think about For example, right, so if you think about features in vision, it is the spatial features, right? So the derivatives and the high frequency features, the edges and things like that. This happens over space. In olfaction, it is over time. That's all I'm saying. Yeah?
Like how many inputs can one projection neuron get from the antenna block? Like is there a limit or is it like?
Okay. So the general rule of thumb in most invertebrates is that only one olfactory sensory neuron type projects to one single glomeruli, and each projection neurons innervates and gets input from that particular glomeruli. So one input channel, one output channel. But that rule is not followed in the locust. So they roughly branch out to and receive input from 10 out of the 800 glomeruli that I mentioned. So 10 out of the 800 channels, input channels that provide information here. Does it make sense? All right. And these responses are highly stable and very reproducible and very odor specific. Right? So what do I mean by that? Now I'm showing you the response of only one projection neuron. There are 3 blocks of trials where we presented 3 different odorants. So when the odor was presented to the antennae is shown here as an orange bar. Okay? The very first trial I'm showing you raw voltage traces showing the membrane potential fluctuations as a function of time. For the second trial, I'm going to assume that the shape and the amplitude of the action potentials do not carry information. So I'm going to digitize this information and rasterize and tell you when the neuron fired an action potential. Is it clear what is plotted in this second row? It is telling you when the neuron fired action potential, that's all. So third trial, fourth trial, there are 7 or 8 trials here. After this block of trials, I present a different odor. Again, I'm showing you the raw intracellular voltages for the first trial, rasters for the rest of the trials. And what you would immediately see is that this neuron was quite silent until the odors come in. After the odor came in, it responded to all the 3 odors, but with different types of activity, different patterns of spike. And those responses were highly consistent across different repeats or encounters of the same stimulus. Does it make sense? Yeah? Okay. All right. So that's one way of doing recording. The other way of doing recording is what we call the extracellular recording. We don't want to record from one neuron at a time. We want to get a handful of neurons, an ensemble of neurons from a single locus brain. So we plunge in a bunch of wires. It's like putting a microphone into a room with full of people, right? So whoever is near to the microphone, their voice is going to be picked up better. Ones who are further away, they're not going to be picked up well. So we can put in a bunch of, this is a neuronexus probes. We're putting it into the antennal lobe of the locust. So we're going to pick up lots of activity, right? And this activity looks something like this. So no longer, this is again, now we are picking up signals which are in the microvolts, hundreds of microvolts, much more smaller, right? And each electrode will pick up signals from multiple neurons. So we look at the activity across all the different electrodes and do spike sorting, as Arindam was saying, to identify and assign a single spike to a given neuron. It is like doing the cocktail party problem with the microphone, right? Saying, okay, this person is speaking, you identify the identity of the person based on the voice signatures. Very similar. So this is a kind of a data set where there are responses from 127 neurons. I have made it into a matrix. So each row here corresponds to one neuron, its activity over time. There are 127 rows corresponding to 127 neurons. On the left and on the right, it is the same set of neurons. The only thing that is differing is the odor that was presented during these 2 trials. So here I'm presenting an ester isoamyl acetate which smells like banana to us. Here is benzaldehyde which smells like almond to us. All right? And what you would notice is that there's some spontaneous activity. When the odor comes in, as soon as it comes in, you can see a lot of these neurons get, the hot colors means high activity, cool colors means low activity. So this hotness that you're seeing over here, over here, over here, these are all the neurons getting active. So you talked about, we heard a little bit about the event-based computing, right? So events are important even in the olfactory domain too. So the on event is amplified here, where a lot of neurons respond with high firing rates. As you continue to present the same stimulus, these neurons either adapt or they drop out. But there are a small fraction of these neurons which continue to respond with a firing rate that is, with a response that is elevated compared to the baseline. So that's what you're seeing over here. And this continues, this particular representation of that presence of the stimulus continues until the odor puff is terminated. After which you have another event, the off event, which is encoded by a different set of neurons. Okay? So this is the on transient, this is the off transient, and this is the steady state response. And each single phase of this response has the information to uniquely identify a specific odorant. Okay? Two different odors, you have a different combination of neurons that participate in the on event and the off event. Okay? So it's very difficult to visualize what is happening in this kind of a plot. I'll show you another way to look at them in a much more intuitive fashion. All right? So another thing to also note is that, so if you look at the sensory input that is coming in from the antennae, that looks something like this. So as soon as the odor pulse comes in, there are a lot of the sensory neurons becomes active. This is the on transient. As you persist, as the stimulus persists, it kind of adapts and settles into this nice fixed activity. It remains there until the odor pulse is terminated, where it exponentially decays back to its original response. If you look at the circuit that is for processing this information in the insect brain, you again have a strong on activity. The adaptation is much more significant. It's much more, you can see this steady state activity is much more closer to the baseline activity. After the odor pulse is terminated, you have this off response. This off event is being emphasized by the neural circuits in the brain, not so much by the sensory neurons. It's emerging out of the recurrent circuits that are present in the antennal lobe. Does that make sense? Yeah?
Does the same thing respond to both on and like when you present the odor?
So the question is, does the same neuron respond both during the onset and on the offset? Yes. Some neurons do, but a majority of them don't. So I'll show you this also. They are two completely different, almost mutually exclusive groups of neurons respond.
So like there are different neurons who respond
Yeah.
to the onset of odor.
Yep.
There might be some different subset which respond to onset.
There is, again, only very few neurons respond both during the onset and the offset. Most of the times, onset neurons are completely different from the offset neurons. Yeah. Okay. Yeah.
Do these insects tend to do some saccading with their antennae, or are they anesthetized and they're just ecstatic antennae right now?
So they can move their antennae when they are freely behaving, but here they are pinned down. The muscles that control the antennae have been removed to stabilize the recording, so we record for several hours, so we don't want them to keep moving. So also, when you're presenting the odor puff, if the antennae is not still, it's impossible to actually present the odor consistently. Yep, correct. Okay, somebody asked about oscillations. Okay, so I just give you one particular slide. So this is a local neuron. We recorded simultaneously the local neuron activity as well as the field potential, local field potential. Okay, the way you think about this is, you go to a cricket stadium, you have the Mexican waves, right? That is the field potential. Lots of people syncing up and actually standing up and down. One particular participant standing in that Mexican wave, that is this neuron in red. Okay, it's a contributor, but again, it's not doing everything. All right. So this is a local neuron. This is the field potential. When the odor puff is presented, you can clearly see there are nice oscillations. These oscillations are not odor specific. All the odors activate very similar kind of oscillations in terms of frequencies, and the relationship, phase relationship between the local neuron and the local field potential is tightly controlled. So it's very phase locked. All right. So let me show you the power spectral analysis. So it is basically showing the frequency as a function of time. At the odor onset, the local neuron oscillates at about 30 hertz, and then later on it kind of adapts and settles into about 15 to 20 hertz. The interesting thing is that the offset, where you still have a strong activity that is going on, does not engage the local neurons, as you can clearly see. There is no activation of local neurons during that particular time period. There is no oscillations during that particular time period. Right? And if you actually do a cross-correlogram between the local neuron activity and the LFP oscillations, you can show that they are tightly phase locked. All right? What I'm telling you now is that during the on period, they synchronize in an oscillatory fashion, the neurons. During the off period, completely different set of neurons asynchronously represent the same information. Yeah?
Just to make sure I understand.
Yeah.
So in this case, the local neuron does not spike.
Does not spike. The neurons in the locust, it does not spike. At most, it fires a calcium spikelet, but that's it.
These local neurons are also specific to certain odor, right?
The local neurons actually integrate input from a lot of the glomeruli. So they do a much more global computation. Yeah, good. Yeah.
So when you said this could be weakly gamma, does the locust have a similar frequency distribution?
No, I think I was just kidding. I was kidding. So it's 30 hertz, so whatever you want to call it. All right? Okay. So maybe we'll teach them meditation and we can push it up further. So, all right. So this is all well known. This has been well established, again, well before my time. All right. So I'm just showing you that we can reproduce some of those basic results. So now going back to how do we perturb these responses, right? So we started Professor Arun's talk. They were using the alphabets and kind of stretching them and making them, distorting them to actually see the visual input being perturbed, right? And the goal was to see how the neural responses encoded that perturbations. So in olfaction, it's very difficult to perturb and change the molecular cues, right? So I'm going to resort to a different technique or a different idea. And the idea is very simple, which we always do. So before I get there, okay, all right. So what do I want to show you? So I want to first ask the question, can the response to a stimulus be perturbed? In other words, can we introduce variations into the neural responses that are elicited by a specific chemical or a odorant? When does this happen? Why does this happen? Is this even principled, and does it help in any computation? The second thing I want to ask is that if it can be perturbed, then how can we achieve invariant odor recognition? All right, those are the two components that I will focus on. So how many of you have gone to a perfume shop and bought perfumes? So a few of you guys. So you smell a perfume. Do you immediately smell the next perfume? What do you do?
Coffee.
Coffee. So what does the coffee bean do? Somehow what you do, so after you smell the coffee bean, you are again sensitized to smell the next perfume, right? Somehow what happened way before, the current stimulus, the next perfume that you're smelling, influences how you are smelling the current perfume. Can I say that? Your stimulus history influences how you perceive what happens next. Yeah? So I'm going to use that particular trick to perturb my responses. I'm going to use odor sequences, right? I'll show you what I mean by that in a minute. So this is again a rasterized plot showing response of only one neuron. So x-axis is time. Each row corresponds to one trial. The tick mark represents one action potential. The color bar here shows when a 4-second puff of any odorant, in this particular case, hexanol, was presented. As you can clearly see across these 10 trials, it's a highly reliable response. Would you agree with that? So it responds to every single trial. There is action potential there every single trial. It's much more reliable during the onset than during the later portions of the trial. Yeah? So now, if for the same neuron, I'm going to present the odors in sequence. So I've presented this particular odor 2-oct, which is also an alcohol, followed by this particular target stimulus, which I'm focusing on hexanol. So in this case, it's identical stimulus. This is the same neuron. Okay? And I'm going to call this first stimulus in the sequence as a perturbing stimulus because I'm using it to perturb the neural response. The second stimulus in the sequence as a target stimulus. Yeah?
How do you precisely switch on and off an odor? I mean, will it persist?
So we have an olfactometer, plus we have vacuum right behind it. So we have characterized the entire setup. It takes a few hundred milliseconds to actually reach the antennae and for it to get flushed out. But that's about it. So we can actually have independent measure of what we are actually, the stimulus we are delivering. Yeah. So this neuron, which was robustly responding to this particular stimulus, is no longer responding to that particular cue. Do you agree with that statement? Yeah. So in fact, I can do different sequences. So now I'm showing you the same neuron. So there's a robust response to hexanol, but I'm now presenting in different 2-odor sequences. The second stimulus in the sequence is always the same target stimulus. All I'm changing is the first stimulus in the sequence. So what do you see? Is there a general rule that you see?
It activates first, then it is not activated.
Yeah, so if the first odor activates this neuron, it no longer responds to hexanol, the target odor. Yeah?
So have you tried hexanol twice?
Yeah, we have.
And
they respond perfectly fine. But even that is a perturbation, right? So even if it doesn't respond, even that is a perturbation. So you still have to recognize a second pulse of hexanol has hexanol, right? So you still have to have the robustness. So even then there's not exactly perfectly, it's not a perfect, you can't guarantee that every neuron that responds to the first pulse is going to respond to the second pulse. Only a subset of them do. So far good? All right. So again, this is one neuron. I can show you the same thing for a different neuron. So here again, this neuron responds to a different odor in the sequence. But here again, the same kind of logic applies, right? So if it responds to the first stimulus, it doesn't respond to the target stimulus. In some sense, you can think of this as the neuron is trying to actually encode if the second stimulus is novel. Okay, if it responds to both, if both these odors activate the same neuron, by not responding to this particular stimulus, it is emphasizing or de-emphasizing the similarity between these two odors. Yeah?
For some of the odorants, there is, like for some of the odorants,
Yeah.
you can see there is highly synchronous activity at the
Yeah.
very beginning of
Yeah.
when you present the example.
Yeah.
So what does it represent? Like too many neurons?
No, again, this is all from a single neuron. Right? It basically means it's getting a strong input, so it's reliably responding to that particular stimulus. This is not as strong as what you're getting over here, so it's a little bit of a diffuse activity. But each neuron has its own free will. They can do whatever they want. Yeah?
Is there a time gap in between two? Like, if you increase the time gap, then it would respond again to it?
So that is true. So if you wait long enough, you can record back the responses. But for up to a few seconds, you will not be able to. And in my case, I want to perturb. Right? So I want to actually make the neural responses as variable as possible. That's my goal. Yeah.
Is that the same time scale for binding and unbinding of these odorants, or those are microseconds, those are much faster?
So binding and unbinding, that's a very tricky subject. We don't know what is going on in the antenna. So the unbinding of an odorant from a receptor is poorly understood. Yeah, all good questions. All right. So this way of looking at things is not going to, again, you're going to be here all day, I'm going to be here all day, we're going to look at one neuron at a time, it's not going to help out, right? So I want to actually come up with a way of visualizing this information as succinctly as possible, preferably a single plot. So to do that, we use what is called as a trajectory analysis. It's a very simple idea. So let's say I'm recording from 3 neurons simultaneously. So let's say this is my olfactometer turning on and off, and these are the responses of those 3 odors, of the 3 neurons. So what we do is that we first take these responses, we rasterize these responses, meaning I don't care about the shape of the action potential, the size of the action potential, all I care about is when the neuron fired. So we've digitized it. We are going to define non-overlapping time bins, and we are going to count the number of spikes in each of those time bins. Okay? So once it is in the analog representation, I can plot it in a 3-dimensional space where x-axis is firing rate in neuron 1, y-axis is firing rate in neuron 2, the z-axis is the third neuron. So in the very first time bin, you have some combination of spiking activity. It becomes a point in this 3-dimensional space. So I can keep plotting every single one of those points as a combination of activity as a 3-dimensional vector in the 3-dimensional space. Right? Once I'm done with plotting all of them, I can link them in the order in which they occurred. So time bin A connects to time bin B connects to time bin C, and I get an odor response trajectory, a neural response trajectory in state space. Okay? That's the idea. I can repeat the same analysis whether it is 3 neuron or n neurons. The steps are exactly the same. So what I do if I have n neurons, this is again schematic. It's not actual data. All right? So I'm just going to show you what I do. So first I'm going to define my time bin boundaries. So once I have my time bin boundaries, I'm going to count the number of spikes. Right? So I'm going to count the number of spikes. And so in the time bin A, I have n spike counts corresponding to the n neurons. In the time bin B, I have n spike counts corresponding to the n neurons, and so on and so forth. N-dimensional vectors are difficult to visualize, so I resort to machine learning dimensionality reduction algorithms, very standard ones. Linear ones, PCA, non-linear ones, local linear embedding, UMAP, whatever you want. All right? Irrespective of the technique we use, we ensure that the interpretation of the data is always the constant. So let's say we pick our favorite technique, let's say it's PCA. We get these 3 axes, which are the top eigenvectors of your response covariance matrix. So now we are ready to plot this high-dimensional vector in a low-dimensional space. Okay? So now, just to illustrate, again, it's a cartoon, not actual data. Just to illustrate what kind of a result you can expect from this kind of an analysis, this blue bar here represents the time bin boundaries I'm using to count spikes. The red sphere here represents activity after dimensionality reduction. All right? So I'm going to move this time bin over time, and you're going to see what happens to the red bar, the red sphere. So before any odor is presented, there are some fluctuations. When the odor is presented, the activity, you activate a unique neurons that change as a function of time is evolving. So therefore, the activity traces a unique path in this state space before returning back to baseline. Right? I can link all these points in the order of occurrence, and I get an odor response trajectory. So again, to recapture this analysis, some fluctuations. Odor is presented, unique combinations of neurons are activated, you trace a unique trajectory in the state space. After the odor pulse is terminated, the response returns back to baseline. That's it, right? So, but it gives you a very quick view of what is going on in the high dimensional data. So I can use this to plot my data from all 85 neurons that I have collected, where I have presented this particular target odor, hexanol, in a solitary fashion. Okay? Solitary means alone. And what I'm showing you, each line is one 85-dimensional vector after dimensionality reduction. These 3 axes are PC axes. Numbers here represents time since odor onset. So I'm just showing you the 4th second when the odor was presented. Okay? B stands for baseline. So the activity kind of gets bigger and bigger. So the length of the vector means more activity. More neurons are responding more strongly. Right? And for about a second, for a half a second to 1 second, it kind of dynamically evolving. This is the on transient. After about a second, second and a half, it kind of settles into some weaker activity close to the baseline. That's a steady state. And that's basically the neural response trajectory for one particular odorant, in this case hexanol, when it is presented alone. So far good? But yeah.
Just curious, so the percentages after the PC are showing what fraction of variance is being captured.
Correct.
So the total is only about 33% or something.
Correct.
So that means like there's still a lot of unexplained variability that I'm not seeing in this.
Absolutely. So yeah, this is again, you need at least about 12 or 13 dimensions to capture about up to 80 or 85%. But in terms of the qualitative dynamics that I want to emphasize and the variability that I want to show, this gives you a good qualitative picture to understand. So the dimensionality reduction is never what you go by as a rule of thumb, right? So you always have to do other analysis to support those conclusions, for sure. But it makes a good visual summary of what I need to say.
So the stimulus was presented for how long?
4 seconds.
4 seconds.
Yeah.
So the plot that you showed before,
Yeah.
hexanol was only shown for a little bit of time, and then the trajectory kept changing long after the.
Oh, that's previous one is just a schematic version.
No, the previous plot.
This one?
Yeah, there.
So this is a cartoon, but I will show you again. I'm just showing you the 4 seconds for a specific reason. There is another 4 seconds that I can show after where the response trajectory changes even more drastically.
Okay, I guess my question is,
Yeah.
that red a bit there.
Yeah.
No, not that. Yeah. Is this the duration of the stimulus?
Correct.
And in general, does the trajectory keep changing long after the?
There are in fact 2 trajectories, one the on trajectory and an off trajectory, and they are completely orthogonal to one another.
And then the off trajectory lasts for several seconds.
For 4 seconds. But then there's still hysteresis in the system tens of seconds later. There's still memory about which odor was encountered, and we can decode the identity of the odorant. So there is a memory about what stimulus is being presented for long after the odor pulse is gone.
Right. So then it would make sense that something that's presented only 500 milliseconds after the first one is going to affect the.
Correct. Yeah. But for my case, in this case, I'm perturbing the response. Stimulus is the response to is hexanol still hexanol now that some of the neurons have stopped responding to that particular odor, right? That's the question I want to ask. Does it make sense? Yeah. Okay. So now I can do the similar trajectories when I present hexanol in different 2 odor sequences, right? So if I do that, this is the response. This red now is a response to hexanol, same odor, but now it is encountered after one of these perturbing stimulus. What you see here is that now it is pointing in different direction. It's evolving in different direction. It's still it's kind of within the subspace of what the other trajectory was, but the dynamics as well as the combinations of neurons activated and how they change as a function of time are all different. I can do this for all the other histories, and you don't end up with one stimulus response trajectory, but you get a family of them. They're all quite variable in which combinations of neurons are active, when they are active, and how they evolve as a function of time. So far good? Okay. So it looks like a mess. There's a lot of variance. Is this variance principle? Is it helping the system achieve anything, right? So that's the next question I want to ask. So I can clearly perturb these responses. What is the computational significance? Yeah.
Sorry, I think.
Yeah. No.
So have you tried doing this in humans and asking whether hexanol, they still feel that it's hexanol if it's preceded by something only half a second earlier?
I've not.
That the brain doesn't think of it as hexanol anymore.
Okay, I have not done it in humans, but I've done it in locusts.
So the locust can tell that?
Can tell you, yeah. We can train them. Pavlovian condition. They can do a behavior. We have a behavioral readout.
So you have a behavioral readout in the locust?
Yeah. Yep. So, any other questions? Okay. All right. So is there any computational significance to this? To understand the computational significance, I'm again going to use trajectory analysis, but slightly differently. Right? So now I'm going to actually show you the response to one of the perturbing stimulus, 2 up now. This is again a trajectory which is very similar to what you have shown before, the entire 4 seconds when octanol was present. This is the response of the 85 neurons to this particular odor, hexanol. As you can clearly see, they are quite different, right? Meaning that different combinations of neurons were activated. Now, what will happen to hexanol response when it is presented in a sequence with 2 octanol? So to understand that, we plotted that one. So all I'm showing you here, the red and the orange trajectories are both responses of the same set of 85 neurons to hexanol, the same stimulus, target stimulus. However, what you would notice is that if I take this red trajectory, if I push it away from the blue trajectories, I would get my orange trajectory. Right? It is made more different compared to the first stimulus in the sequence. Contrast is enhanced with respect to what happened earlier in stimulus history, earlier in time. Does it make sense? What I mean by that is that, let's say we have 2 vectors, A and B. If this vector goes closer to this particular blue vector, that means I'm increasing the similarity between the odors. If I'm pushing it away from the blue vector, that means I'm making it more unique. So when you encounter 2 odors or 2 stimulus in the sequence, the system is trying to, the olfactory system circuit is trying to enhance the uniqueness of hexanol with respect to what happened before. So far good? Okay. I can do the same plots for all the other stimulus histories, and the story is exactly the same. In every single one of these cases, if I take the red trajectory, push it away from the blue trajectory, I will get my orange trajectory. Meaning that in every single case when you have a 2 odor sequence, the second stimulus in the sequence is represented in a way that it enhances the novelty of that particular stimulus with respect to what happened before. Okay? So in olfaction, the context is given in time, not in space. Okay? So maybe we got lucky with hexanol, so we just decided we'll do the same thing with geraniol, with another odor, rose. It smells like rose to us. It has no significance to locusts. So we wanted to make sure that, so Arun said that 2 monkeys is a ground truth, 1 is anecdote. So we just want to make sure that there are at least 2 targets where we can actually get the similar kind of a result. All right. So what I'm showing you here now is blue is still the first stimulus perturbing stimulus. Purple is the target stimulus presented solitarily. The pink is the same target stimulus presented in the sequence. And what you would see in every one of these plots is that if you take the purple trajectory, push it away from the blue trajectory, you will get the pink trajectory. Meaning that whenever we have these 2 order sequences, irrespective of what the perturbing cue is or target stimulus is, we always get contrast enhancement, which is dynamically enhanced with respect to what happened before. I can quantify this by comparing the correlation coefficient, and I can show you that the comparison between the red and the orange or the purple and the pink, you always have a reduction, meaning that the similarity between the 2 odors presented in the sequence reduces when the second stimulus is presented in sequence with the first one. Yeah, there was a question.
Is that contrast enhancement or is the contrast enhancement different for different odor pairs?
The amount of contrast enhancement is greater when the 2 odors actually overlap a lot. Yeah, because they are a lot more common neurons. So they are getting suppressed more, so they'll get pushed further away more, for sure. Yeah, that's a good question.
But then that is not the case if you have 2 similar, like hex sticks.
The second, yeah, that is not the case because you actually still have it slightly contrast enhanced in the sense of with the first stimulus, in the sense that some neurons don't actually respond to that, but it's still very, very similar in response. Yeah.
Then there must be a curve kind of thing, right, where the similarity.
So you can, but if, by knowing which 2 odors are similar to the locust, I'm not going to gain much, at least for this particular study. Yeah. So therefore, we said, okay, all we wanted to do was we wanted to perturb, we achieved the perturbation. Now the question that Professor Ray asked, does the locust even actually recognize the odor as the same odor, given that we are manipulating and changing so many neurons that are responding to that particular stimulus, right? So I need to ensure that they can still recognize and identify the odors in an invariant fashion. Okay, so that's basically what I will focus on next. I'm going to use a behavior. It's a very simple behavior, Pavlovian condition is a paradigm that I'm going to use. Have you heard ring a bell, feed the dog? Right, so just feeding the dog evokes a reflex response, salivation. Ringing the bell does not, but when you pair the ringing the bell with feeding the dog over repetitions, as soon as you ring the bell, the dog salivates, right? So that reflex response gets associated with the conditioning stimulus, the ringing of the bell. Here, I'm going to give an odor puff, give it a grass reward. It's a grasshopper. They like grass. We have starved them, so they will be motivated. Okay, so we do it multiple times, and what they do is when we present a grass blade, they use these sensory appendages close to the mouth called palps to grab that food. Okay, so after training, when we present the trained odor, they open these palps to indicate that they are recognizing that particular stimulus. So in this way, we can actually identify whether the odor still remains the same when we present the trained stimulus in different 2 odor sequences. Does it make sense? Is the logic of the experiment clear? Okay. So I'll show you a quick video of how this looks like. So again, this is to make it quantitative. We paint these palps. You can think of them as giving them a lipstick. So we can track these whereabouts of these palps as a function of time, right? So what you're seeing here is a locust that has been trained in this fashion. So the compound eyes are closed. You can see the green paint there. These are the green pixels. This is the exact location of the green pixels. Here I'm going to plot the distance between those palps as a function of time. So it's sitting quiet. When the LED turns on, the odor puff will be presented. It's a trained stimulus. It's expecting the reward. So it's opening its palp in anticipation of the reward. This continues as long as the odor pulse is there, then returns back to baseline. So far good? Okay. So turns out we can present the trained stimulus and a control untrained odorant multiple times after 10 minutes, 40 minutes, 70 minutes, 2 hours. Once they learn them, locusts have a difficult time unlearning that particular association. Okay? It is a very stupid system. They should be unlearning it faster, but they don't, which is bad for them, but good for our particular experiment because we can repeatedly present the same stimulus multiple times in different sequences and see if they can still recognize that particular odorant. So far good? Okay. So again, this is another way of looking at the same plot. This is the trajectories showing the responses of that particular conditioned stimulus, in this case hexanol. This is the control stimulus. There is no response to benzaldehyde. There is the opening of palp as long as that stimulus is present. Okay, so 10 seconds of stimulus, they open the palp for 10 seconds. For a 4 second of stimulus, they'll open for 4 seconds. For a 750 millisecond of a stimulus, they'll open for 750 milliseconds. As long as the stimulus is present, they'll open their palp to indicate that they recognize that particular trained odorant is present. Okay, that's an important aspect of this particular response. Okay, so now what we did was we presented the same trained stimulus hexanol in different 2 odor sequences. Okay. So I'll start with these three ones because they're easier to explain first. So there's no response to benzaldehyde, citral, or apple, but very clear response to hexanol in each of these cases. So they have no problem recognizing hexanol in these 3 cases. In these 2 cases, they already it's kind of a false positive. So when 2-octanol and isoamyl acetate is presented, they think they are opening the palps. Why? I'll tell you in a minute. Because those 2 odors have a significant neural overlap with hexanol. When hexanol, but the interesting thing is that when hexanol comes in, you can clearly see there is another small increase here. In this particular case too, there's a small increase, much more smaller than over here, but as I said before, they will open their palps and keep it open only if that particular trained stimulus is present. So this basically means they are still recognizing the hexanol. So here, this false start, this false positive is mainly because of the overlap in the neural response between these 3 odors. Okay? So clearly they can recognize the odor, target stimulus, with some false generalization of learning. Yeah?
And when you talked about the representation of the 2 odors,
yeah,
that is in naive, untrained.
That is in naive untrained. Yep, agreed. Yeah?
In the earlier plot that you showed, so the distance between, let's say, IA, not this one, the Earth, where you were plotting the PCA component.
Okay.
So, which, like, if you compare the 3rd row and the 2nd row, which one is more distant? Is it the IA versus Hex or BZAR versus Hex?
So these 2 are quite different. These 2 are quite different. These 2 are quite different in that space.
Plot,
I would not go everything based on the PCA plot just based on the reason that only it captures 30% of the variance, right? So, and I'm also showing you at an angle where I want to make a point. Does it make sense? It's just a plot to make a point, not to quantify. Yeah. But the point here is that this antennal lobe, at least the way we have been, the current dogma is that there's not much of learning related changes that happened in this particular circuit that happens in the mushroom body. So therefore, whatever we are saying should hold in this particular circuit. Okay. So now the question is, okay, how do we go from neural responses that are quite variable to behavioral responses that look quite stable with some false positives, right? So what is the algorithm there? And here again, I'm going to use the same M of n concept. You remember the M of n concept where I said any M out of the n neurons, if it is responsive, I'm okay with it. So let's assume that these are the combination subsets of neurons that are activated by hexanol. When it is presented in different 2-odor sequences, only a small subset of them are responsive. Which subset of neurons are going to be responsive or activated will depend upon which odor sequence it is. We cannot guarantee which neurons are going to be responsive. However, some subset of these neurons are going to be responsive in all these different cases. So as long as I decode information flexibly from this particular subset, any M out of the n neurons being active, I'm going to call that particular stimulus a hexanol, is going to help me decode this particular information. Okay, that's the algorithm. Very simple algorithm, nothing more than that. How would I achieve that? So I'm going to take the neural responses to the solitary exposure of the odorant. All the neurons that were active to the solitary exposure, I'm going to give it a weight 1. All the ones that were inactive or were suppressed, I'm going to give it a weight 0. So this vector is a binary vector, weight vector. There is no free parameter here. So what does this allow me to do? If I take a dot product between the neural activity and this weight vector, it is basically adding up all the spiking activity in these set of neurons that were typically activated when hexanol, the target stimulus, is presented in a solitary fashion. As long as that evidence is above a certain threshold, which is my only free parameter in this particular model, I'm going to say that particular time bin pattern matches with the hexanol response that I typically expect. Okay? So I'm going to do a bin by bin, trial by trial analysis, convert these bin classification output into classification probability afterwards. So it's exactly identical to what I said, M of n concept. Does that make sense? This is the simplest possible algorithm I could think about decoding. Okay? So anyways, so to start with, we did this analysis. So here I am showing you the result of this particular classification analysis. Each of these tick mark here or the raster mark here indicates that particular time bin pattern matches with the hexanol response, on response. Okay? So what you typically see is that whenever the hexanol is present, at the onset you have a strong pattern match with the on responses. Benzaldehyde and citrol, unique odors, they did not have a pattern match. There is some, again, 2-octanol and isoamylacetate. You remember those 2 odors which cross the false positives? They actually have quite a bit of pattern match as well. Okay? So if you convert this particular plot into a classification probabilities, you can clearly see there are nice peaks for octanol and hexanol, isoamylacetate and hexanol, all the different encounters of hexanol, but nothing to benzaldehyde, nothing to citrol. The only thing that throws a wrench into this particular plot is this particular it's small but a significant classification probability that we are getting, pattern match that we are getting with a complex odor which was apple. Do you see that? Yeah, other than that, this particular plot fits perfectly fine with our behavioral responses. You have a false positive, there is a pattern match with the target's template. You have a false positive, again, there is a pattern match with the false template. Here there is a correct negative, whereas we are getting in the neural space, there is a little bit of a pattern match. So how do we fix this? Clearly we are missing something, right? So to fix that, I need to introduce you one additional aspect of this response. So I'm already running over time. So is that okay if I go 10 more minutes? Okay. So I'm going to focus on, so far I have not told you anything about the off responses, right? So question is the absence of a stimulus actively represented? Okay. And so far I have shown these kind of responses where the stimulus is present as raster plots, 2 representative neurons. When you present the odor, these neurons are active either during the transient fashion or slightly delayed and in a sustained fashion, meaning they respond throughout the stimulus duration. Okay? I'm going to introduce you 2 other types where these are the off responses, where the neurons are kind of inhibited when the stimulus is present, but after the stimulus is terminated, they either have a transient response or a delayed and a sustained response. These 4 templates is sufficient to explain all the different neural responses we have in our data set in this particular circuit. So either they are transient at the odor onset or sustained in the odor onset, or they are transient or sustained in the odor offset. So these are selective examples, and this is not one on pathway or an off pathway like vision, right? The same neuron can be on for one and off for the other. Okay? It depends on which odor it is. But nevertheless, there's another way to look at this particular data. So I'm showing you again a matrix of neural responses, again of 80 neurons, a different data set. Darker the pixel, more the activity in that particular time bin. Okay? It's a firing rate plot. I have sorted these neurons such that all the on neurons are at the top and off neurons are at the bottom. What you can immediately see is that when the odor puff is presented, all these on neurons become activated. Do you see that? When they are active, they completely shut down or mostly shut down the off responses, kind of inhibition. After the odor pulse is terminated, these neurons become completely silent, whereas the neurons that were silent so far, they become active. You have completely, very minimally overlapping ensembles of neurons that encode information about that particular odorant, one during the odor onset and the other during the odor offset. Does it make sense? So what does it mean to the response trajectories that I showed you nicely? Every odor has two response trajectories, not one. One during the on period and the other during the off period, and they are orthogonal to one another. Okay? So what does this all mean for behavior? What is the missing piece? How will this help me in eliminating that false positive classification that I had for Apple, right? So turns out the better way to go from neural activity to the palp opening responses is the following paradigm. If there is a pattern match with the on activity, that triggers the opening or initiates a behavioral response. Whereas a pattern match with the off response is necessary or is a better indicator of when the behavior is going to be shut down. So one pattern match activates the activity, other shuts it down. Okay? So if this approach is true, then this should take care of the Apple responses, is what we were thinking. Okay? So now in the same plot, I'm going to also introduce you blue ticks. The blue ticks indicates when there's a pattern match with a hexanol off response. Right? So whenever the hexanol is terminated, you can see a really big blue ticks here, meaning that that's the hexanol off response. Interestingly, what we also see is that this Apple odor, representation of Apple includes both the on and the off responders for hexanol. Therefore, their contribution kind of cancels out each other. Therefore, you don't see a behavioral transduction of that particular neural pattern match. Whereas in other cases, the pattern match with the on response is significantly greater than the pattern match with the off responses. So if you take this and run it to the on-off algorithm, I can clearly predict when I should expect the behavioral response to happen, and that precisely fits what we see in the behavior. So I need two parameters, one threshold for the on activity, one threshold for the off activity, and I'm done. Okay? No fancy deep learning, no complex algorithms. Very simple basic classifier. In both cases, very similar M of n concept. That's it. Any M out of n neurons active. Okay? So hope I have convinced you that the odor recognition performed by sensory circuits is very similar to the way you think about traditional pattern odor recognition. And you need flexible subsets of neurons when the same stimulus is encountered in a variety of conditions to get the robustness or invariance. And the decoder based on a disjunction of conjunction or of ands is sufficient. And then a pattern match with both on and the off responses are important for predicting behavior. Okay? So that's the neuroscience part. There's one small piece that I wanted to share with you was that, okay, what does it mean for engineering, right? So can we make, that does it mean that we can make an electronic nose? Turns out, still today, we do not have good enough sensors. That is the bottom line, right? So the state of art, the gold standard, the state of art is still dogs, their ability to smell, as well as insects and their ability to smell. Therefore, what we asked was, we can't make better sensors. Why can't we use the entire organism as a sensor, right? So have you heard about cannery in a coal mine? No one? Okay. So the idea was that in early days, people used to take a small bird along with them whenever they enter the coal mine. If there are toxic gases present, the bird will pass out, so you get the hell out of there, right? So you're using the bird as a sensor and behavioral readout. But now we can do much more sophisticated thing better than that, right? So what we are thinking was that let biology do what it is good at, convert chemical cues into electrical signal. Once it's in the electrical domain, we can implant electrodes, a bunch of electrodes into the insect brain and take that signal out and use that as a sensor directly. Okay, that's basically the philosophy there. So it's a biohybrid system, if you wish. Okay. So this is the locust. You can see a bunch of wires implanted in the locust antennal lobe. This is the wires coming out connecting to IC socket. They recover back pretty quickly. The surgeries are pretty simple to do if you're trained. And again, they recover back their complete set of behavioral repertoire within about 10 minutes or so. So what we wanted to do now was, our funding came from a certain resource, so we were looking at explosive vapors. do these insects have sensors for something useful, right? So all this thing is meaningful only if they have sensors that have practical applications. So we looked at TNT, DNT, and other explosive vapors. And lo and behold, these guys have sensors for all these different explosive vapors. Some unique combinations of neurons were activated by these different explosive vapors. Okay? So we can take these responses, do a very simple pattern recognition, do very simple algorithms. You can clearly separate the signals out because the sensors were that good. The biological sensors were that good. Okay? So ongoing work is focusing on how to actually get more activity, make as a collaboration with Professor Chakrabarti as well as another colleague of mine, Dr. Singamaneni, looking at better ways to interface with a neural tissue, augmenting the capabilities of the biological system, incorporating the sensory motor components, making sure that ethical and other codes of conduct are maintained, and things of that nature. Okay? But all these things leads me to the challenge slide, right? So what are the research challenges here? So what I said was we can read neural activity out. I never said we can write activity back, right? So to be a truly cyborg or a biorobotic system, you want a bidirectional communication. I don't know about you guys. When I grew up as a kid, the first movie that impressed me a lot was Terminator. Have you heard that movie? Okay. So we are still 40, 50 years afterwards, and we are still not there anywhere close to making a cyborg that looks or behaves or actually achieves the capability of a biological system. So the goal is, the question to you guys as a community is that can the neuromorphic chips provide a nice bidirectional way to interact with, because the claim is that you guys are producing the same language as the biological neural network. If that is the case, then actually taking the output and plugging it into connecting the artificial neural network, spiking neural network with the biological neural network should be seamless and easy to do. And that output coming from your chips should be pretty readily interpretable by the biological network. Is that even true, right? Will that allow us to tap the capabilities, sensory and the motor capabilities of these insects to create a truly cyborg system? And also, can we augment the capabilities of the biology, right? To discuss this and do further things, we'll have a workshop this fall at Washington University in St. Louis. Once I have the logistics finalized, I will share this information with you guys. I hope some of you can consider participating in that particular workshop, where we can continue thinking about this particular fashion, how to integrate the neuromorphic chips with the biological neural networks to actually achieve a truly biohybrid or a cyborg system. I'm going to acknowledge the people who did the work, my collaborators, Srikant and Shantanu. Shantanu is already here. Funding agencies, people who did the work that I showed is primarily from Deb, Rishabh, and Srinath, PhD students and postdocs in my lab. This data set that I showed you is also available to you guys, so I can make it available to you guys. You can use it over the next 10 days if you want. And if you have a better way to do this particular task, please let me know. Feel free to publish it. I'm happy. I'm happy to be proven wrong. I'm happy to be proven to learn something new from you guys. So with that, let me thank you for your attention and happy to answer questions.